Hello, everyone. Thank you so much for this opportunity to share my work. I'm Annie Erickson. I'm a PhD student at Caltech working with Michael Dickinson. And today I'll be telling you about my work on the complete sensorimotor pathway underlying altitude control in flight. And when I say complete, I mean following the flow of information from the sensory all the way to the motor periphery. As humans, we primarily locomote in 2 dimensions. If we're walking along this beach, we can move forward, backward, left, or right. But ultimately, we're stuck to the ground by gravity. Flying animals, on the other hand, like this pelican, are not constrained to walking on a horizontal plane, and instead navigate in 3 dimensions. And they can choose what altitude to fly at. However, the neural circuits underlying altitude regulation remain unknown, largely because it's difficult to record neural activity in flying animals. However, we can use the fruit fly as our model system, as they are expert flyers and fly well in a tethered setup, and we have genetic access to their brain, meaning we can record or perturb their neural activity all while they're flying. So if we're interested in the neural circuits underlying flight control, or really any sensorimotor behavior, the first place we would look in a flying nervous system is this population of neurons called descending neurons. So descending neurons receive sensory input in the brain and project to downstream motor centers in the ventral nerve cord. So VNC is akin to a spinal cord in vertebrates. And through these neurons, this is really the only way that information gets from the brain directly to the VNC. And it wasn't until recently with the development of a large collection of specific driver lines that allowed us to investigate these neurons in greater detail. So we can optogenetically activate these descending neurons using CS crimson, which is a red-shifted light-gated cation channel, all while the fly is flying in a simulated flight arena. And we can record their wing motion with 3 high-speed cameras recording at 15,000 hertz. And the reason that we need to record at such high speeds is that each wing beat shown here takes place in only 5 milliseconds. And we also know that small changes to wing motion can result in large changes to the flight trajectory. So we need to be able to track their wings with high fidelity. So to do so, we developed a pipeline for 3D pose estimation using convolutional neural networks and optimization techniques. And from this 3D pose, we can extract kinematic parameters that we can compare during baseline and activation periods. So 4 of the kinematic parameters that we use to describe wing motion that we can extract from the 3D pose are wing stroke amplitude, which is how far forward or backward the wing extends, wing deviation, which is how far above or below the wing is to the stroke plane, wing pitch, which is the rotation of the wing, and wing camber, which is the deformation of the wing. And we can compare what the effect of activating a specific subset of descending neurons would be on kinematics, overlaid in red. And from these angles, we can reconstruct the wingtip trajectory shown above. I'm just going to focus on the first angle, though, as this was the angle that we saw the most difference within and across lines. And I'm plotting in below the difference between the baseline and activation period, because I know these changes are subtle. So this is the kinematic changes for activating one pair of neurons called DNA10. However, I activated 26 different lines that all project to flight neuropil. And this is a subset of the data here. And what was surprising is that really only a handful of neurons popped out of the screen as having an effect on these kinematic parameters. So on the far right, there's one population of neurons called DNGO2, which our lab and others have studied, and we found that DNGO2 is involved in the yaw optomotor response. However, I'm most interested in this other pair of neurons called DNA10, as it was the only neurons that decreased wing stroke amplitude in our screen. So to further investigate this neuron and see how these changes in kinematics translate into changes in aerodynamics, we can replay the exact wing motion that we recorded during the optogenetic activation screen on a dynamically scaled robot, which we call RoboFly. And that allows us to measure the forces and torques associated with these kinds of wing beats. So the black arrow here represents the stroke average force vector for the baseline period. And in red is the wing beat from a DNA 10 activation. And so we see that activating DNA 10 decreases the upward force and angles it forward slightly. So we would predict from these changes in forces that activating DNA 10 would cause the fly to descend. However, this is impossible to test in a tethered setup. So we collaborated with our colleagues at the University of Nevada, Reno, Floris van Breugel and his postdoc David Stupski, to optogenetically activate DNA 10 in free flight. So here we have a wind tunnel equipped with 12 cameras for tracking the fly over long trajectories. And if a fly flies into a certain area, the lights turn on and we can activate our neuron of interest. And so this is the average trajectory of our empty vector controls. And due to the natural visual features of the tunnel, they descend slightly and then ascend. However, when we activate DNA 10, the flies reliably decrease their altitude. So this really matches our prediction from the RoboFly experiments and showcases how activating just a single pair of neurons can have a robust effect on flight behavior. So next I wanted to see what are the tuning properties of DNA 10. So to do so, I expressed GCamp in our descending neurons and can use a two-photon microscope to record their activity, all while the fly is flying in a simulated flight arena, and I'm presenting different patterns of visual motion. So here is the neural response of DNA 10 to a certain pattern of visual motion, yaw. And I'm showing both the left and right cells as they were both always simultaneously active. So DNA 10 is not very responsive to yaw, but I showed a whole panel of stimuli, and we find that DNA 10 is selectively responsive to progressive, pitch down, and full field translational motion. However, it's not that the fly didn't see these other stimuli, they're actually responding quite robustly. So in the second row, I'm plotting the left and right wing amplitudes. In the bottom row, I'm plotting the combination of these wings. And just to clarify, when I say full field translational motion down, for example, that's what the fly would experience if it's floating upward. So it makes a corrective maneuver and decreases its wing stroke amplitude. However, I was most interested in looking at this pitch response to see if it was truly a pitch response or if it was just responding to the strong downward visual component or down translational component of pitch, especially because I don't have a full 360-degree arena. So to test this, I can present varying widths of downward translational motion, and if it was responsive to pitch, I would expect the neural response to plateau or decrease at a certain width, whereas if it was most responsive to translational motion, I would expect it to continue to increase with increasing widths. And what I see is really the latter, that as I present more downward translational motion, the neural response continues to increase. And I added one last stimuli to really test this further. So it has downward translational motion in the center and upward translational motion in the periphery, which is meant to mimic the translational components of the pitch down stimuli. And I can directly compare it with a stimuli of the same width of downward translational motion. So in orange is what we would expect, but what we see is when we add this peripheral upward motion, the neural response is completely abolished. So further, that solidifies our thought that DNA 10 is selectively responsive to downward translational motion. So now I want to complete this circuit and really look upstream to see where could DNA 10 be getting its visual tuning properties from. So I can look in the connectome, and I see that DNA 10 receives input from a neuron called LPT49, which is a lobula plate tangential neuron. And in a recent preprint by the Reiser lab, they hypothesized that this neuron has a receptive field sensitive to downward motion based on its well-known anatomical inputs. And this would be very consistent with our two-photon results that I just showed you of imaging DNA 10. Next, I wanted to look downstream and see what motor circuits DNA 10 is projecting to. And again, looking at the recently released VNC data, we see that DNA 10 projects to the 1-1, 1-2, and B3 motor neurons. And our lab has actually studied the muscles that these motor neurons control quite extensively. And we know that these three motor neurons' activity are associated with a decrease in wing stroke amplitude. So for example, this is some data from our lab. So the top trace is our baseline wing beat, and the turquoise arrows are the instantaneous wingtip forces. The black arrow is, again, the stroke averaged force vector. And below is what the trajectories that we see when B3 is maximally active. So the magnitude of the force vector decreases, as does the wing stroke amplitude. So again, this is very much in line with what we observed in the optogenetic activation experiments, both in tethered and free flight. So we think this circuit is crucial for flight stabilization. If the fly perceives it's at too high of an altitude or floating upward, it makes a corrective maneuver to decrease altitude. However, it'd also be interesting to see in the future if this neuron is involved in or could be co-opted by circuits for voluntary descent, such as landing on a food resource. And lastly, what I've described so far is really a circuit for descent, but likely there's a complementary circuit for ascent as well. And we have a few candidates in mind, but hopefully future work will fully characterize this circuit. And with that, I'd like to have some special thanks for Johan Melis, who is a graduate student in the lab who pioneered the recording setup, Jaison Omoto, for his help with anatomy and dissections. Again, our collaborators at University of Nevada, my advisor, Michael Dickinson, and the Dickinson Lab, and the Caltech Chen Institute for their funding support. Thank you, and I will take questions.
Thank you, Annie. That's beautiful. We have time for questions. Okay, maybe I'll ask a really quick question. So the idea that it's important for flight stabilization, could you guys do like a gust of wind or some kind of perturbation to sort of induce more of that type of wing stroke? Or?
Yeah, that's really interesting. I think that's probably a very typical circumstance where it would be used, where the fly wants to fly at a certain altitude, a gust of wind comes in, takes it upward, and then it, corrective maneuver. I'm not sure how we would do that experiment. I'm thinking maybe in the 2-photon we do a puff of air, but they won't actually be increasing altitude, so I'm not sure if it would work. But that's a great idea.
Thank you. We have time for some more questions.
Great talk over here. Mm-hmm. I have a question on the computation power of this type of behavior, because I'm working in the field of robotics and reinforcement learning. We know that to build robots that can fly and can do those type of behavior actually is very computationally inefficient these days, right? But for this type of this type of a nervous system, so the computation resource can be pretty low, right?
Yeah, I'm not sure if I'm understanding your question, but I think it's a pretty simple circuit in that, we can basically trace it from photons all the way to the motor output with just a few synapses along the way. Mm-hmm. And well, I would say DNA 10 gets a lot of input from other cells as well, so I'm sure there's more computation going on, but in terms of the computational resources, I'm not sure if
yeah. Right, and for human beings, we have a clear hierarchical control of motion, and but we're only moving in a two-dimensional world. For the three-dimensional world movements, actually the behavior could be more complicated, but it doesn't seem that the Drosophilus need a very complicated hierarchical structure of motor control. So do you think that it?
But I mean, yeah, I guess we don't know the circuit for a voluntary descent, which this could be involved in, but that might be a more computationally intense circuit that involves a lot of more inputs to consider, especially if you need to override your desire to mitigate the optic flow, mm-hmm. if that makes sense.
Yeah. All right, thanks.
Okay, can we have the next person setting up while for the last question over here?
Hey, beautiful talk, thank you. I guess I'm just wondering, if the DNA 10 neurons are always driven just by vision, or maybe like there could be proprioceptive input or the amount of force they feel on the wings as well, and if they could kind of adjust for the trajectory in low light or maybe complete darkness?
Yeah. Hello. Great question. I think, it has many other inputs, so it's possible that it could be driven by, say, a gust of wind, as Anne suggested. But as of now, it seems to be very strongly driven by visual input. So that's all I can say for certain.
Yeah, thank you.
Thank you for your questions.
Let's thank Annie again. All right, next we have Pablo Tano from University of Geneva. Take it away.
Hi, I'm Pablo. I'm a PhD student at Alex Pouget Lab at the University of Geneva, and today I'm going to tell you about our current project. Oh, you cannot see my pointer. Okay, I guess it's fine. So it's predicted representations of compositional primitives. So a few years ago, we trained this artificial agent to rotate a pair of balls. And in the end, after 50 million tries, the agent succeeded in the task. This was very good news, but while I was training the agent, I couldn't stop thinking how much easier it was for me in comparison to do the same task. This is my 5th or 6th attempt. And of course, this is not just humans. In this video, this cat wants to jump on top of the cupboard, and in order to do so, it opens the door, jumps on top of the door, and then jumps on top of the cupboard. I'm not sure if it was the first try, but for sure it was not the 50 millionth one. So in this work, we show that this kind of fast adaptability, by which animals rapidly achieve their desired goals, is made possible by combining two key elements of biological intelligence. Predictive representations, the ability of the brain to predict the consequences of behavior, and compositional primitives, the fact that the behavior that the brain is making predictions about is not just random movements, but rather it is composed of these highly structured and highly predictable behavioral primitives. As an illustration, in a two-dimensional navigation environment, here we separate the trajectory of the rat into two primitives, ρ1 and ρ2. More formally, to start making our model more concrete, we formalize primitives as smooth trajectories, parameterized by their initial position, θ1, θ2, desired final position, desired trajectory shape, and desired temporal extension. Now, the precise parameterization is not important. What is important is that under our framework, a primitive is just a 9-dimensional vector. And adapting behavior in order to achieve a desired goal involves optimizing sequences, compositions of these 9-dimensional vector primitives. And this is exactly what we did. We developed a model that performs this optimization in a sample-efficient manner. Our model has two parts, a predictive representation and an adaptation system. The predictive representation learns to predict the consequences of following sequences of behavioral primitives. So given a state S, the 9-dimensional vector for primitive ρ1, the vector for primitive ρ2, it predicts the intermediate states that will be visited S1 and S2. Then, when a new task is introduced, characterized by some loss function, oh, here's my pointer. Just on time, okay. So, characterized by some loss function L, the adaptation system optimizes the primitive production in order to minimize the loss function. One example of a task could be getting to a goal, for example. So the way this works is, given a state S, the adaptation system proposes primitives to solve the task, ρ1 and ρ2. These are fed into the predicted representation, which predicts the intermediate states S1 and S2. And the loss function is evaluated for S1 and S2, and the process iterates while the adaptation system minimizes the loss function. We implemented the framework with recurrent networks. The predicted representation is just a recurrent network that receives in the input the state and the primitives and outputs the predicted sequence of states. And the adaptation system is just a recurrent network that, given a state in the input, it outputs primitives to solve the task. Now, there are several features of this framework that makes it sample efficient. First, the predicted representation is independent of the task. So it can be trained in a self-supervised manner across tasks. Then the adaptation system doesn't interact with the environment directly. Instead, it interacts with the learned predicted representation. So the optimization of the adaptation system happens in an offline manner without interacting with the environment. So essentially, what we have done is combining a forward model of the world with an inverse model of the world to achieve optimization via offline gradient descent. And this idea is not new. It was introduced 30 years ago by Jordan and Rumelhart. What is new, and is our contribution, is that we have made the entire framework compositional by predicting and planning exclusively in terms of compositions of optimizable behavioral primitives. And it turns out that this compositionality is crucial for the high sample efficiency. So we first evaluated our framework in this two-dimensional navigation task with obstacles. The first task involves getting as close as possible to a goal, it is specified by this loss function, and we deploy the framework in 3 steps. First, we train the predicted representation using 1,000 unsupervised trajectories. Then we introduce the task, we introduce the loss function, and optimize the adaptation system offline. And then the actual tries in the task, the actual attempts that the agent does, are used to fine-tune the predicted representation and make it more precise in the areas of the state space that matter for the task. In this case, it will be around this area, for example. And here are the results. So we see that the zero-shot performance is already quite high, thanks to the offline optimization of the adaptation system, and then performance increases thanks to the fine-tuning of the PR. But the key takeaway here is in the x-axis. So 4 or 5 attempts were sufficient for the agent to achieve expert performance in this complex task. And to show that the task was indeed quite complex because of the environmental physics and the obstacles, so we compare performance with state-of-the-art hierarchical reinforcement learning after 5,000 and 10,000 trajectories in this specific task. So something nice about the model is that the predicted representation is independent of the task. So we can take the same predicted representation that we use for this task and apply it to solve new tasks. And this is what we did. After the first task, we introduced 4 other tasks that involves getting to sequential goals, and maximizing the exploration distance, getting to a safe area and remaining there, and getting to sequential goals while avoiding collisions. In all cases, we see that 4 or 5 trajectories are sufficient to achieve expert performance. So next, we wanted to identify which components of the model are necessary for this high sample efficiency. And to do that, we performed ablation analysis. And unfortunately, I don't have time to go into the details, but the conclusion was that it is the combination of predicted representations, primitives, and compositionality that is necessary for the high sample efficiency. Without primitives, the agent needs to act using immediate actions, which cause transitions from T to T+1. This leads to a very long planning and predicting horizon, which decreases sample efficiency. Without a predicted representation, the model needs to receive feedback by directly interacting with the environment, with reinforcement learning, for example, and this decreases sample efficiency. And without compositionality, the model needs to solve the task using one long and complex primitive, and it turns out that optimizing a sequence of simpler primitives is easier than optimizing a longer, more complex primitive. So everything is necessary. Okay. Our model heavily relies on behavioral primitives. In particular, the predicted representation is trained using primitives sampled from a prior primitive distribution. And it turns out that the complexity of the primitives that the model learns about is a key determinant in the final performance of the model. So here, for example, we show 3 primitive distributions of increasing complexity from straight trajectories, simple, to more complex trajectories with more curves. And of course, learning the consequences of straight trajectories of simpler primitives is easier than learning the consequences of more complex trajectories. So the performance of the predicted representation decreases as a function of complexity. On the other hand, straight trajectories are not very good at navigating around objects, for example, while trajectories that are more complex can navigate the environment efficiently. So we see that the navigation capacity of the primitives increases with the complexity. These 2 factors combine, and we see that the performance of the model in the tasks that I showed before shows a complexity trade-off. Optimal performance happens when the model learns about primitives that are sufficiently complex to navigate the environmental constraints while remaining sufficiently simple to be predictable. Okay, next we applied our framework to this more realistic physics-based simulation environment where the agent controls this ant at the level of 8 joints in order to make it move around. And in this case, we applied the framework at 2 hierarchical levels. At the lower level, the agent simply learns to walk in a desired direction theta. And at the higher level, these walking policies serve as primitives to solve more complex tasks. So at the higher level, a primitive is just a 2-dimensional vector specifying a walking direction, theta, and a walking duration, t. And complex tasks are solved by concatenating and optimizing sequences of these 2-dimensional vectors. We found that after the ant learns how to walk, and after the, after the, Okay. So after the ant learns how to walk, and after the predicted representation of the higher level is learned, the adaptation system of the higher level is able to solve a wide range of complex tasks at the first shot, so high zero-shot performance. These tasks involve walking to sequential goals, walking to a goal while avoiding a hazard zone, exploring the area and coming back, or walking to a safe area and exploring it. It was, of course, very nice to see, after spending 50 million tries in the other musculoskeletal tasks, that we are starting to solve this task in the first shot, after learning a lot about the environment, of course. So as a final remark, I would like to point out that I didn't mention any explicit neurobiological prediction. This is because this is still ongoing work. This is still something we're working on. But as a preliminary comment, note that under this framework, predicting and planning happens in terms of behavioral primitives. And it is crucial for the high sample efficiency to do this, to use compositionality and primitives in order to predict and plan. So if something like this is going on in the brain, then maybe prediction errors and replays should also show this kind of statistical structure. And indeed, there is some recent evidence from Robert Datta's lab showing that in the absence of reward, dopamine activity in the DLS peaks in between primitives. So this is promising evidence in this direction. With that, I would like to thank my advisor, Alex Pouget, and the other members of the Pouget lab, and thank you very much for listening.
Great talk. Time for questions.
Hi. Up here. Hi. Great talk. Thanks very much. Before looking at neurobiological predictions, I was wondering if you were considering making any behavioral predictions.
Oh,
so for a given task, for example, can you predict like the optimal set of primitives that one should be able to observe behaviorally?
Yeah. Like in lines with the complexity trade-off that I presented before, there should be, for an environment, an optimal complexity for the primitives that satisfy that they are both predictable, in the sense that 1,000 unsupervised trajectories or whatever are sufficient to train the predicted representation, and they are also able to solve relevant tasks in the environment. So it's kind of this in-between point that is the optimal under this framework. Yeah.
Question here, and then question over there.
Yeah, so I think 2 intuitive questions. Very nice talk. One is how the loss function might actually be conveyed, because I guess the task learning is basically actually having to get that loss function, which you conveniently get to describe yourself, but the animal has to learn either by observation or exploration or something. It has to map it to the task space. And then the other intuition is about how much time in the offline adaptation learning process has to happen relative to the prediction.
Okay, so the first question, yeah, for the high sample efficiency, it is crucial to have the gradient of the loss function. So to make everything differentiable and optimizable via gradient descent. For example, this is not the case in vanilla reinforcement learning, where the gradient needs to be inferred with a reward signal. There is a way to introduce reinforcement learning in this framework and keep a little bit or most of the sample efficiency. I can show you after if you want. The second question was, can you repeat it? Sorry.
Maybe we don't have time for a second question from the same person. Aaron, do you want to come set up while we take the last question over there?
Hello.
Pablo.
Yeah, really cool stuff. My question was more with respect to how does the agent, in this case, optimize how many primitives are required to do the task? Because in most of your examples, you only showed 2 primitives. Is that a hyperparameter you're fixing the system, or is it something the system can learn that, oh, I should use the minimum number of primitives to perform this task? Is there some sort of regularization for that?
So it should be maybe specified by a high-level controller, kind of a Dirichlet process or something like that. We haven't included that in the model to keep things simple, but I completely agree. Like the number of primitives, or the a priori number of primitives that you need, should be kind of inferred or learned by the system. Yeah.
Thank you.
Yes, thanks. Pablo again. Next, we have Aaron Lanz from New York University.
Thank you. Hello, I'm Aaron, and I am a PhD student in Katherine Nagel's lab at NYU. So today I'm going to talk about my PhD work on looking at working memory and evidence accumulation during navigation. So navigation is not always easy. On the left, this bird is trying to catch a prey that's moving around very quickly. And on the right, this fox uses very sparse and sometimes ambiguous noises in the ground to decide where to leap. And so animals often solve problems like these by using working memory to store, accumulate, and update information over time. So our lab studies how fruit flies find food. And this is also a challenging problem. And this is because the odors that come off of food are dispersed in this dynamic odor plume. And so if you're a fly and you're far from the food, the odor encounters can be very brief and very sparse. And so we think that flies decide where to go to find food, in part by using working memory to collect and store olfactory and wind information over time. And so today I'm going to talk about my attempt to try and answer this question of how does the fruit fly brain store olfactory information in working memory. And so I'll start by showing you this local population and how they persistently encode olfactory information over time. And then I'll dive into the synaptic and circuit mechanisms that are involved in generating those dynamics. So where do we start? Over several years, our lab has identified this group of local neurons in the fan-shaped body, which is the navigation center in the fly brain. And we think this population is really well poised to select which direction the fly walks during olfactory navigation. And so we first found that these neurons integrate information from pathways, from olfactory pathways, and also from wind direction pathways, which means that they receive the right types of information to compute where the direction of the odor is coming. I also showed that if you activate random subgroups of this population, you can cause the flies to walk in a specific but arbitrary direction. So this data is from a single fly where we activated a certain pattern and the fly walks northwest. And so this suggests that activity in this population can select which direction the flies walk. So taken together, we have this sort of model that this local population acts as a locus where it integrates wind and olfactory information to specify which direction the flies walk. And so that's a model. So what's actually happening in these neurons when the flies are doing, when they're navigating? So to test this, a postdoc in our lab, Nick Kathman, designed this closed loop paradigm where a fly is on a walking ball and the wind and odor tube here rotates around the fly in closed loop with its turning, which means the fly can navigate virtually in odor environments. And so this works really great. If you give them a pulse of odor, you can see in pink that they turn and walk upwind in this virtual space. And so when Nick imaged this population of neurons with GCamp, he found that a portion of the neurons would turn on, respond to the odor, and then that activity would actually persist for several seconds after the odor was turned off. And so this is group data showing each line is a distribution of how much that activity persisted for a certain fly for all their trials. So you can see persistence was very common, sometimes lasting up to 10 to 15 seconds after the odor was turned off. but there's a lot of variability across flies and across trials. And so importantly, what Nick found was that this persistence and this variability is really related to the behavior of the fly. So when the activity of this population turns on, the fly will walk straight, usually upwind or near upwind, and that only when this activity shuts down is when the fly deviates from that walking goal direction. And so this leads us to, we can summarize that we found this population of neurons where persistent activity in this population is allowing for this short-term storage of direction information, and we think that the flies actually are using that activity to direct and maintain their walking. And so what I'm going to talk about for the rest of the talk is my portion of this project, which is looking at the mechanisms behind this persistent activity. So to do this, I started by looking at the fly connectome. We were introduced to it in Annie's talk. And so this is a database with every neuron and every synapse in a single fruit fly brain. And I can go in there and find my neurons of interest. And what I found is that these neurons are really strongly recurrently connected with a second set of local neurons. As well, this entire recurrent population are bidirectionally connected with a putative inhibitory neuron. So this was super exciting because recurrent networks with this type of connectivity structure are known to be able to maintain patterns of activity persistently over time. So we thought that this might be what's happening in our system. So to test this, I went to physiology, and I used whole-cell electrophysiology to record from individual fan-shaped body neurons, and I used optogenetics to stimulate olfactory inputs into the system, and I specifically activated ones that would synapse with the entire population, as well as the recurrent partners. And so what I found by activating these neurons was that this activation would generate long-lasting excitation, which could last also up to 10 to 15 seconds. And so we thought if this long-lasting response is due to recurrent excitation, then actually blocking the output of at least this population should disrupt that recurrent activity and prevent this long-lasting excitation from building up. And so to do this, I expressed tetanus toxin in this population of neurons, which prevents synaptic vesicle fusion. And so with that expressed, activating the exact same neurons now no longer cause this long-lasting excitation, but instead mainly cause inhibition. And so we take this to mean that recurrent feedback and recurrent excitation actually plays a really striking role in shaping the activity of this network. So I've shown that recurrent activity is required. So the next question was, how do these recurrent partners actually communicate back onto this first population? So again, I recorded from individual neurons and then stimulated some of these recurrent partners. And I found, similar to what I showed before, that activation of these partners cause a long-lasting excitation that can last up to 10 or 15 seconds. And so these neurons I was stimulating, we know a little bit about them, that they express serotonin. And so we were wondering if sort of slow GPCR signaling could contribute to what is sort of slow and long-lasting dynamics. And so I did the same experiment but applied a serotonin receptor antagonist. And what I found is that the activity became more transient. So you can see after every light pulse, the activity starts to go down, which you don't see without the antagonist. And then after the entire train of pulses, it decays to baseline faster. And so we were curious about this observation. Why would this recurrent network use slow GPCR signaling? What's the purpose? So to understand that in more detail, I generated a model. The architecture of the model was taken directly from the fly connectome. And then so I'd simulate that model using rate-based models and ask how does this network respond to inputs over time? So what I found is that I could tune this network so that if I simulated input, it would respond and show this persistence that could last 10 or 15 seconds, which is very similar to what I saw in my electrophysiology and Nick saw in his imaging. But what I found was that if all the synapses in the network use fast synaptic kinetics, then I'd have to really finely tune both excitation strength and inhibition strength to get the right types of dynamics. So you can see that, so here the color is the decay rate of the network. And so you can kind of see that as this quite narrow red band. But this is in contrast to if I now instead of using fast synaptic kinetics, I add a slow synapse into the recurrent network. And you can see that regime where you get kind of a slowly decaying activity widens quite considerably. And in addition, this network is also more tunable. So if you're in this right regime and you increase the excitation strength, the duration of the persistence actually gradually increases. So we think that networks with recurrent excitation, but also slow GPCR signaling, display robust and tunable working memory durations. So you might be wondering, what is the role of inhibition in this network? And we were as well. And so our first hint came at imaging experiments that Nick did. So in these experiments, he had flies navigate through a virtual dynamic odor plume. And what he found was that the activity of this population would ramp up across odor encounters. So if you've got just like one odor encounter, the activity is quite transient. But if you have many in a row, then the activity starts to ramp and the persistence tends to be longer. And so we wondered maybe when multiple odor encounters happen in a short amount of time, that the network's getting more excitable. And this might be due to some disruption to inhibition. And so to ask if that's possible, at least at the synaptic level, I went back to physiology and I stimulated these putative inhibitory neurons and recorded from fan-shaped body neurons. So we actually didn't know if these were inhibitory, we just had a prediction that they were, so we were quite happy to see that activating these neurons caused this very strong inhibition. And also really excited to see that this inhibition undergoes this really robust short-term depression. And so we thought that maybe this synaptic depression might be a form of disinhibition, where when you have prolonged activity in this network because of many odor encounters over time, that the network will become more excitable because this inhibition is weakening. So to see if this is possible, I went back to my model and I added short-term depression at the inhibitory synapses. And what I found was that a single pulse would cause very transient activity in the network. 5 pulses, you start to see this persistent activity, and then 8, you'd see even stronger and longer lasting activity. And so just by changing the number of preceding pulses from 0 to 8, the decay rate of the network after the odor stimulation was turned off goes from 4 seconds all the way to 14 seconds. But that's not true if inhibition is just static and has no short-term dynamics. And so we think that dynamic inhibition gives these recurrent networks a little more flexibility, where now the duration of the working memory reflects also the history of inputs into the system. So I've described to you how synaptic dynamics and the network architecture can combine to generate working memory in navigation. And so recurrent networks have long been implicated in working memory. And so we think some of our results might have some kind of general insights into how some recurrent networks might have evolved to become more flexible and more tunable. And we're also super excited because we can now leverage the really powerful genetic accessibility of fruit flies to ask how every single component of this network functions. We can ask how those, how the dynamics and how the properties of these cells change with different contexts, different behavioral states, also learning and plasticity. And so those are all things we're going to work on in the future. So I want to acknowledge my lab, especially Katherine Nagel, Nick Kathman, who contributed to the first part of this work, as well as a new collaborator, Bard Ermintrout, and all of you for listening.
Thank you, Aaron. That's beautiful. We have time for a couple of questions. Maybe. There we go.
That was great. Thank you. Up here. Hi. That was lovely. So I'm curious about your optical simulation. I'm guessing you used Orca, but it seemed like you were simulating with the same duration and same intensity each time, right? So if an animal is navigating up towards a source, you're either going to increase the concentration or the filament encounter frequency. So that would make the inputs much more dynamic rather than static. Did you look at that at all?
So we actually didn't stimulate Orca. We stimulated whichever neurons it was like one synapse away. So it was input neurons or neurons in that local network. And I just showed like one type of stimulus at least, the several pulses. I've played with how many pulses. I've played with like the pulse duration. I've done just like single steps. And I'd say most of what I've shown kind of holds across those different stimulus types.
Question here and then.
Nice talk. You showed that the persistent activity.
I don't think I think there's a combination of factors that lead to this persistence. Certainly disrupting the recurrence really like disrupts the network activity, but I think what you're saying is probably also right. Yeah.
When Sophia's setting up, maybe Kenneth can ask a very quick question.
Very quick. So the idea that inhibitory depression plays a role in persistent activity is very interesting. It calls to mind something in the mammalian cortex, which is not just depression of inhibition, but even cell-specific depolarization-induced suppression of inhibition that is carried by the cannabinoid receptors, so that the active pyramidal cells particularly are the ones that lose their inhibitory inputs. Do you think something similar might be going on?
Yeah, it's hard to say because we just discovered these neurons, and I just found out that they're even inhibitory, not that long ago. So these are we have like even sequencing databases that we can try and look for clusters that correspond with these neurons and start to answer these types of questions. Almost certainly I didn't really show some of this data, but there's like different time scales of inhibition in this network as well. There's some fast inhibition, but there's also some quite slow inhibition, which we aren't sure what the molecules are that are causing that. But I think it's a really interesting avenue. Thanks.
Let's thank Aaron again. And finally, in our session, the last talk, we have Sophia Becker from EPFL. Take it away.
Thanks a lot to the organizers for this inspiring conference and for giving me this opportunity to share my work with you today. So let's start by thinking about what we mean when we talk about novelty. Well, except if you're into binging nature's documentaries, probably these two stimuli should be perfect examples of novel stimuli for you. That means that they are new, original, or unusual based on your prior experience. And research shows that we react to these type of novel stimuli actually very differently than to familiar ones. You might have noticed this yourself. Actually, maybe your attention or your gaze were immediately attracted to these images on the left. And based on what we know about novelty-enhancing memory, I guess maybe there's one slide that you will stick to your memory after today. Let's see. So novelty also is very important in guiding our exploration and our decision-making, especially when extrinsic rewards are absent. And these effects of novelty on our cognition and behavior are mediated by novelty signals in the brain that are detected across different areas and then signaled downstream using different neural and neuromodulatory systems. If you want to know more about this, feel free to check out our recent review on the topic. So we've established that these two birds on the left were novel to you, but maybe actually the left bird was a little bit more novel to you, or probably, than the right one, just because the right bird shares a little bit more similarities with other birds that you might know. What this indicates is that novelty is not only a function of how recently or frequently we've seen a certain stimulus itself, but also how similar that novel stimulus is to other familiar stimuli from our experience. And what I want to show you today is how we can quantify this effect and include it in computational models of novelty. To start off, let's think about how we can quantify novelty in experiments. So let's consider this little toy example of a viewing task where we show a sequence of stimuli and we want to predict how novel each of these stimulus is. There are different ways how we can quantify novelty here. I'm going to focus on algorithmic models of novelty that typically define the novelty of a stimulus, for example, the red circle here, as a decreasing function of its familiarity. And that familiarity, in turn, is defined as a count-based frequency. So a ratio of how often we've seen that stimulus divided by how much time has passed. For our specific example, basically the counts of the red circle would increase over the first 3 steps and then stay constant, such that our novelty that we would predict decreases after each observation of the stimulus and then increases slightly because we're viewing other stimuli. So this is the frequency effect in this model. This model is also called count-based novelty and variance of it, and they are extremely useful as long as we can make sure that the stimuli in our sequence here are actually almost perfectly distinct. And there's lots of great studies that actually try this by, for example, using fractal stimuli or categorical stimuli. But we actually get a problem with this model as soon as we want to move to some more naturalistic stimuli, for example, like these natural images here. The problem arises if we think about how to count stimuli that are not identical, but also clearly similar to each other. It's not really clear. Should we count them as the same or as completely different? And then as a result of that question, how should the novelty of a similar stimuli affect the novelty of the kind of paired similar stimulus? This problem becomes even more pressing once we go to continuous state spaces, like for example in exploration problems, because here we not only have spatial similarities, but also infinitely many states, and counting is not only kind of ill-defined, but also really non-informative, because it's very likely we're never going to return to exactly the same state again. So what I want to do today is now to show you how we can actually develop a model of novelty that can account for these kind of stimulus similarities, and by doing that, extend the notion of novelty to more naturalistic and continuous environments. I'm a theoretician, I love toy examples, so let's have another one. So imagine you're seeing the sequence of 3 stimuli, Gabor images with different orientations. I want you to notice that it's exactly one of these examples where count-based novelty is hitting a bit its limit, because we have a continuous state space of orientations here, and we also have significant similarity between these 2 stimuli, and less similarity between the third and the other 2 stimuli here. If we still want to apply count-based novelty, we're basically forced with a few design choices. For example, we could just count each stimulus separately, or maybe we could discretize the space in some way, but then what's the bin size? So there are lots of decisions we have to make, and at the same time, they don't really allow us to capture this difference in similarity between the second and the first compared to the third and the first stimulus. What we can do instead is use a method that we use in machine learning very much to quantify similarities, and that's just put a kernel in the state space. That means, so this also looks it looks similar or simple, so you can imagine it like a receptive field. But what this does, it represents every stimulus in our state space as a function of how similar it is to our reference stimulus here in the center of the kernel, 45 degrees. We can now cover the whole state space with these similarity kernels, and that allows us to express every stimulus as a function of how similar it is to a set of reference stimuli. Okay, cool. So we've talked about similarities, but now how do we get a novelty model out of this? Our approach was to just define the familiarity of a stimulus as a kernel mixture model. So that means as a weighted sum of these similarity kernels here, weighted by a certain set of weights that we are now going to choose such that they encode the kernel frequency, so how often we have seen certain stimuli from that kernel. So in order to make this, for this definition to make sense, we need to choose the weights in a reasonable way. One way to do this is to say our weights should reflect the statistics of the inputs, so we choose them to maximize the likelihood of all the stimuli we've seen up to now. And if we solve this normative approach for our weights, what we obtain is actually, we use expectation maximization for that, we actually obtain a Hebbian learning rule that is local in the sense that it only uses pre- and post-synaptic activity in a multiplicative fashion to update our weights. And this means that we can actually not only implement this model in a neural circuit, but it's also normatively motivated. Okay, so now we've defined our model. Let's see what we actually expect from our toy model. So how much similarities actually modulate our novelty signal in our example. For this, let's first look at count-based novelty and evaluate our novelty prediction for each of these stimuli. What we see here is that the novelty is basically almost the same for all of them. And that is not necessarily unexpected because each of these stimuli are processed separately. So each of them has their own count. What we now do, if we look at kernel-based novelty, we see that the similarity between the first 2 stimuli actually pushes down the novelty that we expect to experience in our second stimulus, while the third stimulus we expect it to be more novel compared to the baseline. Let me quickly run you through a small simulation to understand exactly why this is. So here we can see the weights and kernels that we have initialized to be kind of neutral in a way. We now evaluate the novelty or the familiarity here of the first stimulus. Okay, we get our first novelty point. We update the weights. Because we've observed the state from this kernel, that means we're increasing the weight for this kernel because it's reflecting our frequency. And now we evaluate the second point. we can see that here this point has a higher value on a familiarity curve than the previous one, which just means that its novelty is going to be automatically lower because it shares some similarities that push the novelty signal down for the second stimulus. And if we move on, we do another update and now evaluate the familiarity of the third stimulus, we can see that this curve here, with the familiarity that gives its familiarity, is actually lower than initially, because it exactly doesn't share any similarity kernels with the previous two observations we've made. So as a result, its novelty is slightly higher than the baseline. So we've quantified exactly for this toy example how much we expect our stimulus similarities to modulate novelty signals, and we've seen that there are some qualitatively different predictions we can make. So we expect that there is some impact of similarity modulation, even if we go to maybe neural data. And that's exactly what I'm going to show you next, because so far we had toy models, but we want to see data. So what I brought you here is two open data sets that I used to analyze if our model can actually help us to explain both neural and behavioral data better. The first data set that I'm going to talk about now is from Homan et al. From 2 years ago, and they recorded novelty signals in the mouse V1 while they're basically showing a sequence of different stimuli. Each image that they showed to the mouse was a composition of different gabors with different orientations, frequencies, etc. And they show these kind of images in sequences A, B, C that repeat until in the last repetition, you replace the last image with a novel image, so something that has been unseen before. And then what they do is they record the novelty response as a difference in population response between the response to the novel image compared to familiar images, as they vary different parameters, for example, here the images per sequence. And we do the same. We basically simulate the whole setup with count-based novelty and with kernel-based novelty. If you want to know more about the details, exactly how we scale to this high-dimensional space, you can ask me later. What Homan et al. Observe when they run this experiment is that the novelty responses decrease as they increase the length of the sequence. And if we try to replicate the same finding with count-based novelty, we see actually almost the opposite trend. So it means that we don't really capture some crucial feature of novelty responses in V1 if we only count. Instead, if we use kernel-based novelty, so include similarities in our analysis, we can replicate exactly the same qualitative trend, that novelty decreases as we increase the length of our sequence. We have similar findings also for other features. So when other parameter variations that Homan et al. Do and that we replicate, and I'm not going to show them now, but this suggests that kernel-based novelty can actually help us to capture V1 novelty responses and specifically predict which of them might be due to some kind of representational similarity modulation here. Okay, so we expect our novelty signals to be modulated by how much similarity we have in our sequence of stimuli. So the next question that's obvious to ask is there a tractable effect also on behavior, especially novelty-driven behaviors? And to find out about this, I looked at a second data set, which is by Rosenberg et al. They let mice explore a labyrinth freely in absence of rewards. So basically, I'm looking at the period before they received the first water reward, and then model the mouse behavior as intrinsically motivated reinforcement learning agent that either follows count-based novelty or kernel-based novelty. What we find if we do this is that the reinforcement learning agent that seeks kernel-based novelty actually manages to explain mouse exploration much better than if we only look for count-based novelty. So it seems like there's also some impact of similarities, in this case more spatial, that influences the behavior of our mice in this task. Okay, to sum up some take-home messages, I've shown you how we can account for stimulus similarities on novelty signals by extending this framework to use similarity kernels. Actually, I want you to notice that this framework is consistent with count-based novelty when similarities are absent, but we can also apply it when there's similarity structure or when their state space is continuous. We can update this novelty model with a bioplausible in terms of local pre-post learning rule. And this model helps us to explain both the novelty responses in mouse V1 and the spatial exploration of these mice in a labyrinth. With that, I want to thank my amazing collaborator Alireza and my very supportive supervisor Wulfram and the whole lab at EPFL for their encouragement and support. And yeah, thanks for your attention. I'm happy to take any questions now or later.
Thank you, Sophia. Time for a couple questions. Here.
Thanks for that great talk. I was curious, do you consider any relationship between novelty and aesthetic value, or novelty and preference or likability of the sensory objects?
Okay, so if I understood the question right, is if I consider any connection between the actual subjective value of the representations that might be already associated, is that correct?
Either the preference or likability of the object, or the aesthetic value of the object. It's close to what you mentioned, but more specifically about these two, likability or aesthetic value.
Yeah, that's actually, I like the question. So it's true that the complexity of the stimulus would be encoded in the kind of representation that we have so far. So in a way, this model is modular in the sense that you can put it on top of any representation. So if we have a very complex stimulus, we would expect that it's encoded more richly, such that our similarity kernels would be also more densely populated in that part of the state space. Okay, thank you.
One quick question. Yes.
That was really great. I was just wondering how you defined the kernels in the Rosenberg experiment modeling you did. And does it matter if you use like geodesic distance versus just like Euclidean kernels, like Gaussians in the environment?
Yeah, great question. So at the moment, the first approach that we had was to define kernels that summarize areas of the maze that the mouse is exploring. So it's a bit less sort of place field oriented. So so that would be based on the distance in a binary tree, because the labyrinth has a shape of a binary tree. So that was the first approach. Now what we're testing, and the results are not completely done yet, is to see if we can reproduce a similar preference for kernel-based novelty if we actually use place field shaped kernels. So this is current work, and I'd be happy to discuss more, but we don't have the results yet.
Very cool.
Thank you, Sophia. Let's thank all the speakers from this panel.
Thank you.
Coffee.